Bismillahirrahmanirrahim and Assalamualaikum to everyone who watching the video SCL Presentation. Today we as a group 3 we are presenting about muscular system. This group consists of 4 group members which are Muzaffar, Khairina, Balqish and also myself, Syaira. Before we start, I would like to say sorry first because this presentation had to be in virtual due to the coronavirus that attack our country nowadays. So because this is our first time doing the presentation in virtual, there will be some problems throughout this presentation and we really sorry about it. But we really hope you guys can catch up and understand what we want to present today. Okay, based on the title that will be given, which is Muscular System, you guys can refer my textbook page 736. Now I will start first, and I will start with the introduction. So as an introduction, we already provide a picture of muscular system at there. You guys, it look complicated, right? But don't worry guys, because in syllabus we just need to memorize a few parts. For example, you guys just need to take note and pay attention for the something that already bold in the media textbook. Okay, for the first word that had be bold at there is tendon. So what is tendon? Tendon is the skeletal muscle are attached to the skeletal by band or fibrous connective tissue. In a simple way, tendon is something that attach muscle to the bone. We already know in Form 5 we study about it, right? So your tendon is attach skeletal muscle to the bone. So skeletal muscle. The information about skeletal muscle is when the muscle contract, there will be shorted. But you guys need to remember, muscle can only pull, they cannot push. That's why they work in antagonistic pair. Antagonistic pair means, sebagai contoh saya bagi, tangan kita kan ada tricep dengan bicep. So bila tangan kita lurus, bicep akan relax and tricep akan contract. Dia kena berpasang-pasangan sebab bila dia tak berpasangan, tendon memang tak boleh, skeletal muscle memang tak boleh buat dua perkara dalam satu masa, macam relax dengan relax. Dia kena relax dengan contract. Itu memang itu memang skeletal muscle work in our body. Then we go to the tightening. Tightening is occur when the muscle contraction build. So definition of tightening is maximum sustained muscle contraction. It occur in the body muscle whenever skeletal muscle are actively used. Then we go to the tone. Tone macam tadi tightening when the muscle contraction build, but for tone it different because it occur when muscle need relax. So the definition for tone is situation when muscle relax, some fibrin contraction. It is important to maintain posture. For example, I give to you all, if all the fibrin in the muscle in certain our body, for example neck, trunk, legs, when all of these relax, the body would collapse. That's why we need to tone. Tone. Macam contoh, bila saya tidur, bila saya berdiri memang akan ada muscle yang relax, tapi kita punya neck takkan relax waktu tu. Kalau dah start relax, kita akan collapse sebab kita tak ada posture yang maintain. Then we go to the microscopic anatomy and physiology. Okay. At here we have sarcolemma, sarcoplasmic reticulum, myofibrin, and scaromeryx. And so for the scleroderma, is the definition is the cytoplasm of meiotic. It form a transverse system. Then we go to the sarcoplasmic reticulum. The definition of sarcoplasmic reticulum is the equivalent of the smooth endoplasmic reticulum in meiotic. It consists of expanding portion of modified endoplasmic reticulum. This is for storage portion. This is for storing site for calcium ion Ca2+. Ca2+ it will need at the nanti Mus akan terang dengan lebih detail pasal yang ni. Myofibric is the definition of myofibric is the contractile portion of muscle fibrils. What the important of myofibric is it run the length of the muscle fibril. Is band responsible for skeletal muscle treated appearance. So the last one is sarcomeryx. The sarcomeryx is the tempat protein filament in myofibric within contracted unit. I think that's all. This picture will explain more detail by Muzaffar later. I think. Thank you.
Today I want to talk about the study filament model of muscle contraction. First, we're gonna look at the basic structure of the muscle. You guys can check on the slide number 4. The muscle consists of the tiny strand called the myofibrils. The myofibril is then squished together to form a larger structure called the muscle fiber. The muscle fibers are the actual cell of muscles, which consists of mitochondria and nuclei. They are covered up on some sort of plasma membrane called the sarcolemma. Then the muscle fiber form the bigger structure called by the name of bundle of muscle fiber. The bundle of muscle fiber is then attached together to form a larger structure called the muscles. We're gonna zoom in at the myofibrils. You guys can check on slide number 5. The myofibrils are divided into segmented. Each segmented are called by the name of sarcomeres, which is the contraction unit of muscles. Within the sarcomeres, there are two tiny protein strands, which is called by the name of myosin and actin. And in the sarcomeres, you guys can see there's the Z line. The Z line structure, the function of the Z line is to attach the actin filament, which is the thin filament, and divide each sarcomeres. And the myosins is the thicker strand is placed in the middle of the sarcomeres. You guys can check on the area that is in the sarcomeres, which is the air band, which is considered the area that consists of myosin and actin overlapping to each other. I band is only the area that is contained on the actin. The H band, the area that contains on the myosins. Okay, during the contraction, the actin filament slide past the myosins. This causes the I band to be shorter and the H band to be nearly disappear. Okay, although the actin slide past the myosins, but the myosin who actually do the works. In the presence of the calciums, myosin break down ATP and form a cross bridge between the myosin head and the actins, causing the myosin head to pull the actin closer to the sarcomeres. So the myosin head let go the actin and grabs the further down the actin and pull again the actins into the middle of the sarcomas until the process continue, until the contraction is completed or the sarcoma is completely contracts. Okay, notice that the myosins or the myofilaments do not contract or expand. They just slide past each other. Alright, for the next slide, I'm going to explain about the usage of ATP in the muscle contractions. You guys can refer on the slide number 7. ATP provide the energy for the muscle to contract. Although the muscle cells contain myoglobin who store oxygen, but the cellular respiration cannot provide the energy instantly for the muscle to contract. So the muscle fibers rely on the creatine phosphate, who anaerobically react with ADP to produce ATP and creatines. When all the creatine phosphate depleted, the ATP is made then produce more energy for the muscles contractions. So I think that is all from me. I will pass on the presentation to Balqish. Thank you.
Okay, next I will present about muscle innervation. Muscle are stimulated to contract by motor nerve fibers. Nerve fibers have several branches, each of which at an axon terminal in close proximity to the sarcolemma of a muscle fiber. Synaptic cleft is a small gap that separates at axon terminal from the sarcolemma. Axon terminals contain synaptic vesicles filled with the neurotransmitter acetylcholine, which is ACH. When nerve impulse traveling down a motor neuron arrive at axon terminal, the synaptic vesicles release ACH into synaptic cleft. ACH quickly diffuses across the cleft and binds to receptors in the sarcolemma. Now, the sarcolemma generates impulse that spread over the sarcolemma and down T tubules to the sarcoplasmic reticulum. The release of calcium released from the sarcoplasmic reticulum causes the filaments in sarcomeres to slide past one another. Sarcomere contractions results in myofibril contraction, which in turn results in muscle fiber and finally muscle contraction to a neuromuscular junction and has initiate response. It is removed from the junction. When the enzyme acetylcholinesterase ACHE breaks down acetylcholine, muscle contraction ceases.
Hello, my name is Nur Khairina and I will be presenting about the role of calcium in muscle contraction. So, we have learned that the calcium ions come from the sarcoplasmic reticulum. But how does it actually function in muscle contraction? I will explain about that in detail, but first we will take a look at this figure. So let's take a look at this figure. The figure shows three components of muscle contraction, which are the actin filament, the tropomyosin, and the troponin. So first of all, we will look at the actin filament. The actin filament is actually composed of two rows of twisted actin molecules. Next, we will look at the troponin. Troponin is actually where the calcium ions will be attached. And lastly, we will look at the tropomyosin. The tropomyosin is the green thing that looks like a thread. It actually winds around the actin filament, and in interval, troponin are between them. The calcium ions will bind to the troponin. This will cause the tropomyosin to change its position, and this will also cause the actin filament to change its position as well. Upon doing this, the myosin binding site is exposed. The myosin binding site is actually where the myosin head will bind during the muscle contraction. So the first step of the muscle contraction is actually when the ATP binds with the myosin head. The myosin head looks like the golf club. So when the ATP binds to the myosin head, it will hydrolyze the ATP because the myosin head is actually an ATPase. It will be hydrolyzed into ADP and phosphate group. Once the ATP is hydrolyzed into ADP and phosphate group, it will activate the head so that it can bind to the myosin binding site at the actin. So next, the ADP and phosphate group remains at the myosin head until the myosin head attaches to the actin site to form cross bridges. Cross bridges is actually the attachment between the myosin head and the actin. Once they are attached, the actin and the myosin head, the ADP and the phosphate group will be released and it will change their position due to the power stroke. Power stroke is actually the act of pulling the actin filament towards the center of the sarcomere. Now that the myosin head is empty, ATP will bind back to the myosin head. This will cause the cross bridges to be broken and the myosin head will actually detach from the actin filament. The ATP will hydrolyze into ADP and phosphate group and the whole cycle will repeat again until the actin is fully pulled towards the center of the sarcomere and until the muscle is fully contracted.
Okay, in the act of presentation, our presentation. Sekarang waktu untuk Q&A segment. Okay, for the Q segment, I will conduct the Q&A section. Okay, we will provide 5 questions, and if you guys want to answer it, just comment. And because this presentation had to be in virtual, right? Okay, for the first question, what is the function of myosin filament in the muscle contraction process? Number 2, what happens with calcium when ion calcium 2+ bind with the troponin? Number 3, according to the sliding filament model that be explained by Muzaffar, when muscle contract, what will happen? And number 4, what is acetylcholine line? And the last question, this is actually not in our presentation, but I think there's any video can answer it, for especially Danisha and Amma. So the question is, how long does it take to recovering when the tendon is torn? I think that's all. Thank you for hearing our presentation. Thank you. Okay, sekarang bila Khairina dah habis, kita akan buat Q&A session. Okay, in this session kami sediakan 5 question, and siapa-siapa yang nak jawab tu boleh komen ataupun cara yang sendirilah untuk jawab soalan ni. Okay, soalan pertama, what is the function of myosin filament in the muscle contraction process? Number 2, what happens when ion calcium 2+ bind with troponin? Number 3, according to the sliding filament model which Muzaffar just explained just now, when muscle contract, what will happen? Number 4, what acetylcholine line? Okay, and the last question, actually last question is not in our presentation, but it's the common knowledge.